last piece on the parietal cells. And remember yesterday we were emphasizing the role of the G protein coupled receptors as being the transmembrane proteins that would bind to those three major players on the parietal cell. And we talked a little bit about the histamine released from the ECL cells. We talked a little bit about the gastrin being released from the G cells, and we've just begun to touch on the acetylcholine being released from the postganglionic parasympathetic neurons. But in all of those cases, we're going to see that these are G protein coupled receptors. And I want to finish that story with a couple of clinical implications, and these are things that we see in our everyday lives. And if you go to the grocery store, you can buy antacids, and one of the major antacids is cimetidine. Cimetidine then is, in fact, a H2 histamine blocker. So effectively, we'll use the unique character of those histamine receptors, those G protein coupled receptors, on the parietal cells, and when we take the cimetidine, we'll block that H2 receptor. More commonly these days are the imiperosols, and the imiperosols work a little differently. The imiperosols are actually a pump inactivator, and what the imiperosol does is it binds to the active site on the hydrogen ion potassium antiporter. And when it binds to the active site, it permanently inactivates it. And one of the reasons that the advertisements for the molecule for drugs like Prilosec and Nexium, you only have to take it every 24 hours, and that's because that permanently inactivates the pump. These are both important for acid reflux disease that we talked about yesterday. Then what we're going to talk about is moving the chyme into the small intestine. Yesterday we spent a lot of time generating the chyme, talking about the anatomy of the stomach. Now what we're going to do is move the fluid in the stomach into the small intestine. It's going to move across another sphincter. Again, this will be smooth muscle. It's going to be a circular smooth muscle at the interface between the stomach and the small intestine. And we'll talk about how this pyloric sphincter, when it gets the right signal, opens up, and the smooth muscle of the stomach can squirt, 30 to 50 mils of fluid from the stomach into the small intestine. That will give us an opportunity to spend a little bit of time on the structure of the small intestine. We'll talk about the tunics. We'll see the tunics are very similar to what we saw yesterday for the stomach. Then we'll talk about the regions of the small intestine, being the duodenum and the jejunum and the ileum. And we'll just touch on those in passing. And I'll remind you that most of what we're going to be talking about on Monday is going to be in the jejunum and the ileum, the jejunum and the duodenum, where most of the absorption will take place. And then finally, what I want to talk about here are the so-called villi. The villi in the small intestines, this term villus means finger, and these villi in the small intestine are multicellular structures, but they become important multicellular structures, so we need to spend time talking about them. They're really these multicellular finger-like projections, but they're multicellular in that they are formed from a simple epithelium of enterocytes. Basically, we've been calling all these epithelial cells the GI tract epithelial cells, but now these are the enterocytes. And the enterocytes now are going to be forming the structure called the villus. Now, one of the interesting things about the villus is the enterocytes have this typical organization for epithelial cells. They have an apical surface and a basilar surface. And we'll see then that right beneath the basilar surface will be located blood vessels. And these blood vessels are unusual in that they are part of the hepatic portal system. If you take an anatomy course, you'll spend a lot of time talking about the hepatic portal system. But for in here, all I want to emphasize is that there are these specialized blood vessels that are going to have these tiny capillaries that reach up right beneath the enterocytes in the villi. And what we'll see then is in these blood vessels, we can now, through these blood vessels rather, we can transfer digested amino acids and glucose. That is, the final products of breaking down food are going to be transferred into the blood vascular system in these capillaries right beneath the villi, and that blood is then destined to the liver, and that's mostly what we'll talk about on Monday. But there's another character here right beneath the enterocytes, and this is called the lacteal. And the lacteal is actually lymphatic capillary. And we'll see on Monday that the lymphatic capillaries here are essential for taking up lipids. And you remember I mentioned just briefly, that our handling of lipids is different than our handling of water-soluble nutrients, such as the glucose and amino acids, which we'll talk about going into the hepatic portal vein, but the lacteal will be taking up lipids, be sending them right back into the blood. And we'll talk a little bit about that. But most of what we want to talk about today is once we get the chyme into the small intestine, there's going to be a level of hormonal control that we want to talk about. Again, this is going to be back to secretion. And the first set of cells we're going to talk about are the so-called S cells and the I cells. The S cells and the I cells are modified enterocytes in the intestinal mucosa. And these S cells and I cells are actually kind of monitoring cells. The S cells are going to monitor for a change in pH. Remember, the contents of the stomach will have a pH sometimes as low as 2. So this very acidic contents of the stomach is going to be squirted into the small intestine. These small intestinal cells, the enterocytes here, don't have a heavy mucus layer of them like we saw in the stomach. And what's more is all the enzymes that are going to be doing the digestion in the small intestine have a pH optimum of around 7.2 to 7.4. So one of the first things we have to do when the chyme comes into the small intestine is to neutralize that acid, bring the pH back up around 7. That neutralization is carried out by a combination of the S cells here in the intestinal mucosa and a set of cells in the pancreas called pancreatic duct cells. We're going to see that the S cells release a hormone called secretin. It's a peptide hormone. That peptide hormone then goes from the intestinal mucosa through the blood vascular system and then finds its way to the pancreas. And in the pancreas, it binds to receptors, G protein coupled receptors, on the basolateral side of the duct cells. That signals these cells now to secrete bicarbonate. They're going to look like parietal cells turned upside down, where the parietal cells generated hydrogen ion and pumped the hydrogen ion to the lumen of the stomach. Here we're going to see these pancreatic duct cells are pumping bicarbonate into the lumen of the pancreas that empties into the small intestine. And the bicarbonate then is going to be able to neutralize the hydrogen ion. Then the other set of players in the exocrine pancreas are the cells known as the pancreatic acinar cells. The pancreatic acinar cells then are going to have on them receptors for the molecule released by the I cells. Where the S cells are monitoring for pH, the I cells are monitoring for lipid and protein. When the I cells see lipid and protein, they send out a hormone called cholecystokinin. The cholecystokinin then, as one of its major roles, will then bind to these pancreatic acinar cells. And they will then signal the pancreatic acinar cells to begin releasing a variety of digestive enzymes. They'll be releasing chymotrypsinogen, they'll be releasing trypsinogen, they'll be releasing lipases, they'll be releasing amylase. So the acinar cells are involved with releasing the digestive enzymes. The duct cells are involved with releasing bicarbonate. But these are working together then to begin the process of digestion in the small intestine. Then, if there's time, we'll spend a little bit of time discussing two major phases of digestion in the small intestine. One of them will be the bulk phase. And the bulk phase is going to be in the middle of the tube. And the bulk phase is going to be where the peristaltic action of the smooth muscle is going to be squishing fluid back and forth and back and forth. It's like a washing machine in there. But any of you who are engineers who have had some fluid dynamics will remember that regardless of how much activity you have in the middle of a tube, up against the wall, there's hardly any movement. And that's true in the small intestine too. So the fluid up against the wall, right against the epithelium, moves hardly at all. So we have about a 30 micrometer thick layer, which is called an unstirred layer. So where in the bulk phase, we're going to be squishing things back and forth and back and forth and doing a lot of the digestion of great big proteins into smaller molecules, great big molecules into smaller molecules. So we'll be converting proteins into peptides, and we'll be converting starch into maltose. And then the final digestion is going to occur along something known as the brush border, which will be extensions of individual enterocytes and the enzymes as transmembrane proteins. And we'll talk about that probably on Monday. But where I want to start here is just to finish the story quickly of where we were yesterday. And we talked about the parietal cell. And we described the parietal cell as being a cell, an epithelial cell in the gastric pits, part of the mucosa. And its major function was to have out on its apical surface this transmembrane protein, which was a hydrogen ion potassium ATPase. We said accompanying those proteins out of the apical membrane in these blind alleys of membrane were potassium channels. And we talked about the idea that the potassium channels would simply permit the potassium to run down its concentration gradient and end up in this blind alley. Once the potassium was in the blind alley, then it could be bound to the hydrogen ion potassium ATPase, and there'd be an exchange. It required ATP, it required energy. But the net effect was to allow for a hydrogen ion to leave the parietal cell and to, in exchange, bring a potassium ion back in again. And then we finished with a brief discussion of the hormonal control of these cells. And we said that there's a G protein coupled receptor in the basolateral membrane. And this G protein coupled receptor bound the molecule gastrin. The gastrin is released by another epithelial cell in the stomach mucosa, the G cell. And that gastrin then would go all the way through the blood vascular compartment, work in a truly endocrine fashion, and bind to the G protein coupled receptor. The other player was, I'll put this one in blue, I don't remember what I used yesterday. And this was the H2 histamine receptor. And the H2 histamine receptor responded to histamine that was released from the enterochromaffin-like cells. The histamine here functions mostly in a paracrine fashion. And that's simply to say that the enterochromaffin-like cells are nearby. They're in the neighborhood, and they're releasing histamine from the basolateral side. That histamine is binding to this receptor and initiating the cascade of events for this G protein coupled receptor. And finally, I'll put this one in red, this is a G protein coupled receptor, which is a muscarinic receptor. And this muscarinic receptor responds to acetylcholine released from postganglionic. Parasympathetic neurons. This, of course, is acetylcholine. The acetylcholine binds to this receptor, and again, we're working through G protein coupled receptors. Now, if you get down into the weeds of this, these all work slightly differently, but for our purposes, these are all impinging on increasing hydrogen ion release. So there are effectively three parallel, mutually reinforcing pathways that lead to an increase in hydrogen ion release from the parietal cells, and thereby increasing the acidic content of the stomach, thereby lowering the pH. And the last piece on this, what I want to talk about are these sort of clinical implications. Cimetidine is basically an H2 receptor blocker. It exploits the idea that we don't have many H2 receptors outside of these parietal cells. So the cimetidine binds to the receptive site that normally binds histamine. It basically blocks the action of histamine, and in itself, cimetidine has no effect on the G protein coupled receptor. So effectively what we're doing is the cimetidine is blocking. It's a blocker of the histamine. So it turns off this branch of the increase in acid. It's fairly effective, but it's not as effective as the omeprazole. And these are the ones we see advertised on TV. These are the ones that are Nexium and Prilosec. And the way these guys work is they inactivate irreversibly. Always want spell check on the board. Inactivate irreversibly the hydrogen ion potassium ATPase. And that's terrible. That's closer. Irreversibly, yes. All right. What it does is it goes into the transporter and inactivates the transporter. Now you take that out of the circuit altogether. And the reason that omeprazole only has to be taken every 24 hours or so, and that's part of its shtick when you listen to the advertisements, is that it fully inactivates these transporters. And the only way you can get reactivation of the release of the hydrogen ion is through the upregulation of genes to make more of the hydrogen ion potassium ATPase. So what we're saying here is that one of the advantages of the omeprazole is you take out the pump. And when you take out the pump, you simply aren't going to pump any hydrogen ions. And then you'll wait several hours before you get a significant addition of these proteins in the membrane, so you get a significant amount of acid generated. Now, the reason we use these, of course, is the gastric reflux disease that we talked about yesterday. We have heartburn continually because that sphincter, the esophageal sphincter is not working properly, and we're getting reflux of the stomach contents into the esophagus. And it's unpleasant and it's dangerous. And so what we do is we take these drugs to sort of ameliorate that problem. There have been some cases where there's a suggestion, at least, that by blocking those transporters, that you have some higher risk of some bacterial infections from foodborne pathogens. So you just don't have that acid in the stomach contents to lower the rate of cell division among the bacteria. But let's remind ourselves where we are. There's the esophagus, the stomach. And here is the esophageal sphincter. And down here is the pyloric sphincter. So where the esophageal sphincter was regulating access to the stomach, the lumen of the stomach, now the pyloric sphincter regulates access to the small intestine. Well, remember the contents of the stomach is known as chyme, and it has a pH of less than 2. So you don't want to move all of this contents immediately into the small intestine. So when the pyloric sphincter opens, it allows only about, I think it's 30 to 50 mils of chyme to enter the small intestine. Well, it's worth taking a moment here and looking at to look at some of the anatomy of the small intestine. So over here, I'm going to talk about anatomy of the small intestine. I'm going to first talk about the so-called tunics. These are really the layers. And we spent some time yesterday talking about the tunics of the stomach. These are going to be very similar. I'll start here with a kind of squiggle. This is meant to be the mucosa. This is the intestinal mucosa. It, too, is an epithelium and areolar connective tissue. As before, immediately beneath that will be this layer of submucosa. And then outside the layer of submucosa will be a layer of smooth muscle. Now, in contrast to the stomach, where we had 3 layers, a longitudinal, a circular, and an oblique, we only have 2 layers here. We have one that's running parallel with the axis of the small intestine, and then there's another one that basically runs around the small intestine. And this is meant to be smooth muscle. And then beyond the smooth muscle, out here is now going to be that layer that we talked about before. This is the layer of the mesothelial cells. Known as the serosa. So fundamentally, we have the same kinds of layers that we described before. We have a mucosa, a submucosa, a muscle layer, and then the mesothelium. Now, I need to spend just a minute here talking about parts of the intestine. So the small intestine is divided into 3 regions. The first is the duodenum. Second is the jejunum. And the third is the ileum. The duodenum is actually fairly short. It's only about 12 inches. Now, there's some meaning in this term. The duodenum is derived from the Latin duo, decum. Duodecim means 12. And the story is that there was some early anatomist, probably in the late Middle Ages, early Renaissance, who was doing gross anatomy, and they started looking at the intestine. And they saw a division between this region they called the duodenum and the jejunum, and they measured it by finger widths. And so they said, okay, there are about 12 finger widths between the duodenum and the jejunum. Well, we know that's 12 inches now. That's one fat-fingered anatomist. Not many people have 12 or 1 inch wide fingers. I mean, even Donald Trump doesn't think his hands are that big. Then the next region is the jejunum, and the jejunum comes from the Latin, means empty. And you may have heard this when you were studying for your GREs or your SATs. And often we use this word too in everyday language, where we talk about someone being jejune. They mean they're empty, without character. And it's derived from the idea that when they were actually doing the initial anatomy, that they would always do this on individuals who had been executed. And so they'd been dead for some time. And so when they got to the jejunum, it didn't have anything in it. So they said, it's the empty region. And to this day, we call this the jejunum. And the ileum is the third section, and it then empties into the large intestine. So those are the major regions, and we're really going to be focusing on these two. This is where most absorption occurs. Now what I want to do, now let's look at a couple of slides first. So this first slide shows the overall structure of the tunics of the small intestine. And that's chaos in here. Okay, here it is. And so what we're looking at is just a pretty picture of the crude drawing I have on the board. And it's showing that the lining of the intestine is thrown into these folds. And the folds are then further folded into these structures called villi. Now let's walk from the mucosa out. Here's the mucosa. There's a submucosa shown here. Then the muscular layers, one layer of circular, one layer of longitudinal, and then out to the serosa, which is formed by those mesothelial cells. And they're showing the blood supply here coming into the small intestine. What I want to do now is I want to take this little region, it's just this one little finger-like structure, and blow that up. And that's shown in the next slide. And we're going to spend a lot of time on this structure, and I'll try to make some crude drawings of it. But this is a pretty picture of a villus. And there's some really important aspects to this villus. You notice this finger-like projection is multicellular. That is, each one of these dividing lines here represents an individual enterocyte. So there are hundreds and hundreds of thousands of cells in each one of these villi. It's a multicellular structure. These epithelial cells or enterocytes are lined up in such a way that the apical regions face the lumen of the intestine, and the basilar regions now face this underlying series of blood vessels. Now, the blood vessels are, in fact, a branch of a particular blood vessel called the hepatic portal system. The hepatic portal vein runs through the intestines, and all of that blood in the hepatic portal vein is destined to find its way to the liver. And we're going to talk about that on Monday. But what I want you to see now is we now have these fine capillaries as branches of the hepatic portal vein. And these fine capillaries now are going to be strategically placed right beneath the basilar surfaces of the enterocytes. And what we're working our way toward here today and to the beginning on Monday is going to be that the intestinal contents are just on the other side, on the apical surface of these epithelial cells. And then we're going to be able to use epithelial transport to move glucose and amino acids in much the same way we saw in the proximal convoluted tubule, and move those molecules from the lumen of the intestine across the epithelial cells and into this extracellular space. Now that extracellular space, you'll notice, is right next door to the endothelial cells of the hepatic portal vein. So we can now, just as we saw, if you put your phone away to appreciate it, then what you find is that these molecules will move from the lumen of the tube across the epithelial cells and across the endothelial cells and into the blood of the hepatic portal system. That blood then is going to be carried to the liver, and that's where we're going to further take care of glucose molecules and amino acids. And we'll talk about that in more detail. But what I want you to recognize right now, though, is that we have these finger-like projections, hundreds and hundreds of thousands of them. And these are all composed of epithelial cells called enterocytes. Yes?
So when you have the lipid going across into the lacteal, does it go directly to it, or is it going through the blood capillary and then get pressed
Okay, so the question here is about lipids, and I wanted to hold this off, but I'll go ahead and deal with it. We're going to find that when we talk about lipids, well, let me do it another way. We're going to find that when we talk about the products of starch, which would be glucose, and the products of proteins, which would be amino acids, if you put your phone away, I would appreciate it. Please don't give the eye roll. So those will be water-soluble molecules that are going to move across the epithelial cells directly. They'll move from the apical surface to the basal surface and then out into the extracellular space. But you're right, lipids are different. Lipids are going to be taken up as monoglycerides and fatty acids. They'll be taken up into the enterocytes. And then, of all things, they'll be repackaged as triglycerides in the ER. And then, once they're repackaged in the ER and Golgi, then they're mixed with proteins because the lipids aren't very soluble. So you mix them with proteins that are a little more soluble. Then you package them in vesicles and release them as something called chylomicrons. The chylomicrons then are released into this space. But the chylomicrons are so big. They're like close to a micron or more. They're too big to move across the endothelium of the hepatic portal system. So instead, they move into the lacteal, which is a lymphatic capillary. Turns out the spaces between the endothelial cells lining the lymphatic capillary are bigger. And so you have better access for these big honking, what they call chylomicrons, to get into the lacteal. And those are then swept in the lymphatics all the way back to the subclavian vein and dumped into the subclavian vein. And we probably won't get to it, but then finally what will happen is that lipid, once it's in the blood, will then be drawn out of the blood and put into adipocytes. But again, at that level, you break them down into monoglycerides, actually into fatty acids, move the fatty acids into the adipocytes, the fat cells, and reassemble them again. It's remarkably baroque, the way lipids are dealt with. And it would seem to be energy inefficient, but there's so much energy in those molecules that we are willing to pay that price. So hold on to that, and I'll hope to get to it on Monday. So let's imagine now what's going on with when this chyme at such a low pH moves into the tube of the small intestine. So basically, if we imagine this structure, we're squirting about 30 to 50 milliliters of chyme into the small intestine. These epithelial cells forming the mucosa are not covered with a heavy layer of mucus as we saw in the stomach. So they're labile to proteolytic digestion. They're labile to damage at low pH. What's more is we're going to see the enzymes that will be released from the pancreas have to work at a pH level 7.2 to 7.4. So we're going to find then that we have to regulate the pH of the contents of the small intestine. So any questions to here? Because we're now going to start into hormonal control. So let's take the upper panel. And I'll try to introduce the players here as being the small intestine, the epithelial cells of the small intestine, part of the mucosa, and a character we've talked a little bit about in here. We talked about the exocrine pancreas, but we're going to sort of dive down a little deeper into the structure of the pancreas. And so the major players here that we're going to be talking about will be, let's make the list. First one will be S cells. So the S cells in the intestinal mucosa. There are going to be another set of cells called I cells in intestinal mucosa. There are going to be a set of cells called duct cells in the exocrine pancreas. These, of course, are going to be epithelial cells. And the fourth character here is going to be something known as a acinar cell. The duct cells and the acinar cells are both part of the pancreatic epithelium. But they, in fact, do different things. These duct cells of the exocrine pancreas secrete bicarbonate, whereas the acinar cells of the pancreatic epithelium secrete digestive enzymes. The S cells in the mucosa secrete secretin, and the I cells of the intestinal mucosa secrete cystokinin. Now, we normally abbreviate this as CCK, but there's actually some meaning in this term. This is bile, sac, move. The reason it has this name is it's a peptide hormone that was originally demonstrated to bind to smooth muscle in the gallbladder and cause contraction of smooth muscle in the gallbladder. And so it's bile sac move. But we now know it has a really other important role in that this cholecystokinin, or CCK, is going to lead to the release of digestive enzymes from the acinar cells. So let's see if we can put this whole story together. So there's the pyloric sphincter. Sitting in here, sort of as a brown structure, kind of tucked up in here, is a structure known as the pancreas. And the pancreas has a tube that leads into the small intestine. Now, if you take an anatomy course, you'll see that this comes into the common bile duct, and the pancreatic duct joins with the bile duct. But don't worry about it. What I want you to recognize is that the pancreas has a layer of epithelial cells that's continuous into the small intestine. Let me do this a little differently. Now, there's a continuous layer of epithelial cells, and I drew it too small, but if you can see that there's this continuous layer of epithelial cells. Some of them are the epithelial cells of the pancreatic duct. Others are these cells back here, which are epithelial cells of the pancreatic acinus. These are referred to as the acinar cells. So the two major players here in the pancreas, in the pancreatic epithelium, will be the duct cells and the acinar cells. But meanwhile, down here in the small intestine, let me make this a little bit bigger. Again, among the epithelial cells of the mucosa will have cells which I'm going to label here as modified enterocytes. Now, an enterocyte is just a specific term for an epithelial cell in the small intestine. So these enterocytes are modified enterocytes. I'm going to put one of them in here, excuse me, and put one of them in here and make it red. And I'm going to label this as an S cell. The S cell monitors the pH of the intestinal contents. Remember, the chyme coming in from the stomach has a really acidic pH. And so when these S cells recognize a high hydrogen ion concentration, low pH, what the S cells do from their basolateral surface is they release secretin. Secretin is a peptide hormone. The receptors for the peptide hormone, now let me blow this up a little bit. Where down here, I'm going to try to blow up a cell which is going to be one of these so-called pancreatic duct cells. It's an epithelial cell. It's forming the lining of the duct of the pancreas, so it sits on a basal lamina. It has a basilar surface, it has an apical surface. And as a component of the basilar membrane, there's this transmembrane protein, which is a secretin receptor. It's a G protein coupled receptor. What the G protein coupled receptor does when it binds to the secretin is that it increases the release from the apical surface of bicarbonate. And it increases the release from the basal surface of hydrogen ion. And in fact, this cell uses the same molecule that we talked about yesterday. Here in the cytoplasm of this cell is going to be carbonic anhydrase. And when the secretin binds this G protein coupled receptor, it again increases the activity of the carbonic anhydrase. And the carbonic anhydrase here is using carbon dioxide and combining it with water to generate a hydrogen ion and a bicarbonate ion. Same story as we saw yesterday in the parietal cell, same story as we saw in the erythrocytes in the lung. But now what we see different today from what we saw yesterday is the hydrogen ion goes out the basolateral side. So it goes back out into the blood vascular compartment, because remember, down along this basilar side will be a series of capillaries. This is a capillary. So the same blood vascular system that's delivering the secretin is now going to take up the hydrogen ion. Meanwhile, the bicarbonate is being released from the apical surface. And remember, the apical surface is facing the inside of the tube of the pancreas. So that means the HCO3 negative is now free to move down through the pancreas and into the small intestine. And the opening of the common bile duct is very close to the pyloric sphincter. So now what we can do is that when the chyme comes into the small intestine, we can now, from the pancreas, release bicarbonate and take the pH of the intestinal contents from down around 2 or 3 back up to about 7.2 to to 7.4. So we've buffered all that hydrogen ion using bicarbonate. And now we have basically neutralized the hydrogen ion with the bicarbonate. So we now have a contents in the range of about 7.2 to 7.4. No. Okay. Are there any questions to there? So the next thing I want to do before you guys leave is that the other character being released from these cells here will be digestive enzymes. So the acinar cells are going to be releasing pancreatic amylase and pancreatic lipase and trypsinogen and chymotrypsinogen. Now, in the last minute, the signaling here is going to be where the eye cells, again in the mucosa here, let me put this in purple. These eye cells, the eye cells are monitoring for lipid and protein coming out of the stomach. And the eye cells, when they recognize lipid and protein, these eye cells release CCK. The CCK then goes into the blood vascular system and will then bind to these epithelial cells, the acinar cells in the pancreas, and that leads to the release of these digestive enzymes. Next time what we'll do is we'll talk about how these digestive enzymes are working in the bulk phase and the final digestion is in the brush borders. I thought I had plenty of time.